Welcome to our second episode of the 2021 Speaker Series. My name is Lale Welsh, the CEO at the Neuromuscular Disease Foundation. Thank you for joining us. Today, we're going to be speaking with Rich Horgan. Rich got his Bachelor of Science degree at Cornell University and later went on to get his MBA from Harvard Business School. He has been nominated as the 30 under 30 at Forbes, which is very impressive. And I believe a couple of other accolades that I don't recall right now, too numerous to mention. Without further ado, I'd like to introduce Rich Horgan. Thank you for joining us, Rich.
Yeah, thank you so much for having me, Wally. Really appreciate the time and the opportunity. And to all the folks listening and who will listen, thank you for taking the time to do this. Before I dive into my presentation, I think just a little bit of personal reflection in the sense of, I started this mission because the traditional way of drug development wasn't something that was going to help my brother and my family. And so given that sort of desperation, if you will, it became important to figure out how can we change the existing system, or how can we modify the existing system so that it better serves our patients, and most importantly, the ones that we hold dear. And in my case, that's my brother. And so today I'll go through a presentation just kind of outlining that journey of what we've done at Cure Rare Disease and what we're starting to do at NDF, and encourage questions. I'm not entirely sure how they're coming in, but I believe we have 15 minutes set aside for Q&A, and I think Angela will be supporting that effort. So today we'll talk over, in terms of an agenda, two broad points. The first is going through what we've done at Cure Rare Disease as an N=1 or single patient therapeutic development for my younger brother who has Duchenne muscular dystrophy, as well as how we can take that same framework and apply it to other gene therapies, other monogenic or diseases, genetic diseases that are caused by mutation one gene for GNE Myopathy. And so before we dive in, excuse me, I'd like to share a short video. It's about 4 minutes long, and it really sets the stage for the story that I'm about to tell.
In a way, rare disease is a misnomer. It's a lot of people.
My name's Rich Horgan, and I'm the founder of Cure Rare Disease, the nonprofit that's developing customized therapeutics for rare disease, and our first target's Duchenne muscular dystrophy. I have a brother with Duchenne muscular dystrophy.
I'm Terry Horgan, 23.
Terry was diagnosed with DMD when he was 3. Duchenne's a rare disease, a disease that saps one's muscle over time. My mom's 3 brothers had it and all passed away from it. It's 100% fatal.
My uncle Bobby had the same disease. And when my grandmother found out, she got on a train, went to Boston with Bobby all by herself, and the doctor at the time said, "Go home and love your son." That's all. That was it.
We didn't want to just take Terry home and love him. We wanted him to have a life.
Watching your brother not being able to do the things that other kids are doing, not being able to play ball or go to prom, strikes home. There's not a lot out there for Duchenne.
We're not even on a clinical trial because there are none.
The trials are geared for younger children. They take all the information from the older boys over the years, and then they're kind of like pushed aside.
It's easier to get results from, younger kids that are still walking. Not a lot of for non-ambulatory, so we're kind of left out. Forget about us type thing, we're still here.
The pharmaceutical companies need to make money, and how can they make money from an N=1 case, from a rare disease patient? We need to change the paradigm.
Every individual with Duchenne muscular dystrophy is slightly different from the other one, which complicates to say, okay, how do we get a molecule that fits all these different sort of permutations and combinations that is Duchenne, and more broadly, other diseases.
Angela and I looked at it and we strategized going, what approaches could we take to actually help Terry instead of what approaches exist?
What's unique about this particular moment is that those tools are getting to the point where we can actually use them, not just for making drugs to treat tens of thousands of people, but we can actually use them to set up home workshops, build solutions for diseases that don't have, say, the financial backing of a huge industry.
What we chose to do from early on was to house this in a not-for-profit, because the idea is that we can move more nimbly because we're funding a group of collaborators.
This allows us to move with a clarity and a speed that traditional industry may not be able to do. Working with patients who are very sick, there's a real incentive to move fast, to actually say, I've got something that could potentially help you, not in 10 or 15 years, but next year.
Give them a little bit more incentive to, put in the extra hours.
More so than ever, that kind of investment is beginning to pay off.
Tim, over the course of 10 months, developed a customized therapeutic for a little girl. A year and change later, she's still with us. This isn't fantasy, it's not sci-fi, it's happened. We did this. This has been done.
Specifically for Terry, it's starting to pay off now, and we're seeing restoration of full-length dystrophin.
Then we can do this. We can win. We have the plan. We have the team. We're executing. We've made achievements that have never been made before in the field of Duchenne.
Treating patients like Terry and translate that to other patients with different ranges of DMD mutations and also different genes. That way we can actually scale.
What we learn from every patient doing this customized approach empowers the next one and the next one, and we move quicker.
That's the opportunity that exists in the field to exploit, and that's why we're so excited to have this foundation supporting this kind of work.
We need every bit of help we can get, and every bit of help matters here. Every dollar, every cent, every person who wants to come support, every corporation who wants to partner that's doing something that will make a difference, that will save a lot of lives. Now's the time. My brother's 23. Time's not on our side here. And to have his trust and faith that we can do this, and shown so in blood and sweat and tears, is powerful. We're on the edge of pioneering the development of customized therapeutics, and there's hope.
So that was hopefully a helpful intro into our conversation today. Terry, now 2 years down the road, as I'll share with you, we have developed an effective gene therapy for him, tested that in his cells and animal models. We've had a conversation with the FDA through a pre-IND meeting, and as Laude mentioned earlier, we're planning to dose him later this year. And so there's a lot of tie-in into that story into our conversation today. As you saw, Tim's approach for Mila really began to set the framework where we could develop gene therapies or spur the development of gene therapies for patient populations that were too small, too limited, or simply not yet enough sort of quote unquote raw material for industry to become interested. And so by building a team of collaborators and developing something that could affect positive change for Terry, we realized and we said, wait, there's actually much more here than we thought initially. And there's a couple different ways to take this approach, whether it's a single patient who has a deleterious mutation that's resulting in a disease of some sort, or potentially it's different than that. Potentially, as in the case of GNE myopathy, how do we develop the early stage research, the mouse models, the therapeutic constructs, the preclinical data? How do we as a group, as an organization, how do we help to enable and bring those into fruition so that we can go further faster? And as you can see, with as it pertains to Terry's drug, his was, as I mentioned, customized to his unique mutation. It was successful in upregulating the missing protein, both in a dish of his cells, as well as in the experimental animal models that we moved on to subsequently. And for those of you out there who may not be as familiar with what a gene therapy is, the question of gene therapy really comes down to delivery, especially for muscle diseases. And the question of how do we deliver these gene therapies systemically throughout the body? Muscle, I believe, is about 40% of our mass. And so actually getting therapeutics to individual myoblasts or myotubes, individual muscle, is actually quite difficult. And so to do that, we use technology called AAV. And AAV stands for adeno-associated virus. The little 9 after it means the different type of AAV. And so this sort of early stage of gene therapy has ushered in our first generations of AAV. And AAV, you'll hear AAV9, AAV8, AAV RH74. They're different permutations, slightly different forms of AAV that all come from a relatively simple virus. Not simple, but relatively common virus. And so as society progresses into the next generation of gene therapies, we'll start to see different numbers next to AAV. AAV, for instance, 9.2 or 9.23, something like that, just as an example goes. But AAV is not a perfect tool. And it's important to understand as a patient community, what does AAV mean and what do the implications of using AAV lead to, especially for patients? Given that AAV is a virus, there's a significant potential for an immune response. An immune response can be along a wide range of different levels. A minor immune response that can be dampened with the use of corticosteroids, for instance, or a larger immune response. Now, in the early days of gene therapy, this wasn't necessarily as well understood, but what we're seeing now is, oops, sorry, I was just trying to get that little window out of the way there. What we're seeing now is that there's a better understanding of what predisposes an individual patient to have a more serious immune response to the administration of AAV. And we're working on technologies to better dampen that, to allow AAV, or a FedEx truck, if you use an analogy, to deliver that package being the therapeutic contained within the AAV, to the place it needs to go. You know, you can think of AAV as the FedEx truck, the package inside as the Amazon package, and your house being the muscle in the case of muscle wasting diseases. Some challenges that still exist with AAV that we're just starting to wrap our collective minds around include the ability to redose. So it's challenging for people to get the same virus twice. And we learned a lot about getting viruses twice through this past year and a half with COVID. But more or less, the massive amount of viruses that's used to deliver AAV systemically throughout the body, whether it's for Duchenne, GNE myopathy, limb-girdle muscular dystrophy, so on and so forth for muscle diseases, it's a lot. Generally, these things are dosed on a per kilogram basis. So you'll often see the formula or the dose being a really big number. For instance, 1 × 10⁻⁴ and that's the number of particles viral genome per kilogram. So you can imagine if you take your weight in kilograms multiplied by that 1 × 10⁻⁴, that is the pure number of viral particles that are entering the body. And so the body reacts and goes, oh boy, that's a lot of virus. We need to develop an immune response. And what the hope is that the therapeutic will get in, do its work, and then over time in the ensuing days, weeks, and months, the body will then mount a response to not allow that same virus to get to itself twice. In the case of a therapeutic use of AAV, as we're doing, the idea is that, do we need to redose? How long does the therapeutic last? These questions are on the minds of researchers and at the forefront of thinking of how do we go from our early successes in generating gene therapies to the reapplication of gene therapies down the road, if and when they're needed. But more on that and encourage questions and conversation on this. It's a very complex topic. So I've tried to just present some of the high level thinking on that. Moving forward, what I wanted to do here on this slide was just give you an idea of the speed that we had to move at. It's not necessarily speed just because we want to go fast. It's speed that's dictated because Terry is wasting away from this disease. And for those of you who may not be as familiar with Duchenne muscular dystrophy, or DMD as it's commonly abbreviated, DMD affects only boys for the most part. 99.9% of patients are boys generally. The disease starts to manifest in early single-digit years, so 5, 6, 7. Mom or dad will notice something's not quite right, maybe an abnormal gait, missing growth marks in early childhood development. And that'll usually query a visit to the doctor's office, and the doctor will run a blood test or a genetic test, and that's generally when the diagnosis is made. Clinicians often do it on the back of elevated CK or creatine kinase, which is a hallmark of muscle damage. And so after that, there can be a genetic test run to better define the exact mutation. The gene itself is very long, and so mutations occur at different points along that gene. In my brother's case, he has an inactivating mutation, meaning that the beginning of the gene doesn't work. Think of the gene as a recipe book. Dystrophin has 79 pages, and so like if you're making a cake with 79 steps, first of all, it's kind of a crazy cake, but second of all, without the first page, the body just doesn't really know where to start. And so sometimes the body will get clever and try to start further down the gene. But for the point of this conversation, it doesn't matter as much. The idea here is that genetic diseases are often very, very complicated, and patients are very different from one to the other. But here, we had the idea to start this in fall of 2018. And at the beginning, we really didn't have a great idea of where this would all go. At the time, and continues to be really the bleeding edge of science, in the sense that developing a drug for 1 or 10, 100, 1,000 patients is something that's relatively new. But in taking that biopsy, we were able to characterize the mutation, understand the genetics and the molecular biology behind it. Our wonderful team at Yale with doctors Monkle and Angela Leck spearheaded the charge to actually develop a therapeutic construct, something that would do the therapeutic activity, result in the production of more protein, put simply. And by spring of 19, that was accomplished. And then we moved into an animal dosing study right before COVID broke out in March of 2020. And then we met with the FDA in October of 2020 through a pre-IND meeting. And the idea here is that we've done a significant amount of work before that meeting, and we wanted to get the FDA's guidance and perspective on their thinking about this technology. The specific technology we used for Terry was a CRISPR-based technology. And while the technology for this conversation doesn't matter as much, it's more so the framework that's of interest for this conversation. We wanted to be sure that we were closely collaborating with the FDA to be sure that, as we got further and further down the development path, and as cost and time increased, we didn't want to be blindsided by something that the FDA had a purview on, that we could make those changes earlier on in the development cycle, rather than execute on a number of experiments, get the feedback that, oh, we wanted a different animal model, or we wanted a different type of analysis done, and then have to redo those. So early conversations with the FDA were especially helpful for us. Now we're hitting on a really, really exciting point, the manufacturing of the human grade drug. This is the same drug that was used in the mouse studies, but is a higher purity and quality than that drug used in those mouse studies, is about to be complete. So in the end of this month, what's called the GMP stands for Good Manufacturing Processes. If you've ever seen that terminology, or human grade, you can associate it to, will be done. We will then do our final safety study, which will start in July, and then we'll compile that all together and aim to dose later in 2021. So it's been an exciting journey. And this journey, at least for me, was not one that was known to me. For the folks listening, I was very much so in your shoes, where I'm watching my brother deteriorate over time, not much to do about it, but feelings of powerlessness, hopelessness. These things are very real, and these things are very scary. And so I really resonate with the folks out there who are impacted by these genetic diseases. This is a really, really challenging journey. And I think for me, I don't come from a particularly wealthy background, relatively, I guess normal guy with a relatively middle class and normal family. I really needed to figure out how I could take this idea that was really my brother's best shot and transform it into reality quickly. And so to do that, I leveraged the opportunities that I could leverage the networks of Harvard Business School and Cornell to get conversations and open doors that would have otherwise been closed to me. Leveraging media opportunities. How do we take these opportunities with Forbes or Business Insider or the Boston Globe and tell the story of what we're trying to do. I think above all, you know, if nothing else comes of this journey, we're not at the end of it yet for Terry. You know, if nothing else, I hope this idea of rapid lean drug development is a framework that will help many other people down the road. And I think for me, it's not, you know, I'm not a scientist by background. I'm very fortunate to have a great team of scientists, some of whom are with us here today, who have really rallied around the cry to try to reinvent or add to society's arsenal in terms of how we can develop drugs quickly, safely, and effectively in a way that's slightly different. And now with COVID, depending on where you are, I guess, becoming less of an issue, we can see that society is quite capable of doing this. You know, a vaccine in under a year, a bit around a year, is really impressive. And so how do we take those lessons, which we've really painfully learned, and apply them to diseases that aren't as, don't have the same level of awareness as a COVID? How do we take these lessons and apply them to GNE myopathy? And so moving into the next part of the conversation is where I want to get to how we're doing that. And so what you can see here is an overview of the same framework that we used for my brother. This idea that we could rapidly design or utilize existing therapeutic constructs here on the far left. And then with a therapeutic construct, I should say again, is just the entity that goes in the AAV, the delivery vehicle, the FedEx truck. The analogy here is this is the Amazon package. This is the workhorse that does the therapeutic activity, either eliminating a protein if it's a storage disorder, or upregulating a protein if it's a lack of a protein causing the disease. And in the same time, we need to find an animal model. Does one exist? In the case of GNE myopathy, fortunately, there are a few animal models. No animal model is perfect, and the FDA understands that. The FDA looks and says, we understand that there may not be a perfectly representative model of this disease, but as long as there is a model of some sort, whether that's a cellular model of patient cells, or whether that's an animal model that reflects characteristics of the disease, muscle loss or other functions, then we can use that to start to generate the data that we need to convince the regulators and convince ourselves, most importantly, that this therapeutic has legs, that there is activity here that's exciting, that we think this could do benefit for patients. And a lot of the time is really spent in actually de-risking it. Once we've got the animal colony and the constructs identified, then we can take those constructs and package them into an AAV. We do this through usually research grade material. Again, if you remember back a few minutes ago, GMP was the human grade, but for animal models, we use lower grade material simply because of the cost and time associated with that. We'll get to manufacturing in just a few minutes. But with a quick experiment, with a quick in vivo or in life, in mouse model is what it translates to, we can get an idea of the therapeutic activity. Do we see the upregulation of an interesting of the protein of question, or a down regulation, depending on what disease we're going after? And this really helps us to really start to build the data package. In vivo data is great. In addition, testing the construct on patient cells is also a helpful exercise to get us an idea on the cellular level of, do we see therapeutic activity? Do we see, you know, the restoration of what we're looking for? Now, with those two things, where we go from here is that conversation with the FDA, right? So we've done some in vivo experiments, we've done some in vitro or in glass or in patient cells as it pertains to this conversation. And oh, by the way, FDA, here's the work we've done. Here's our plan for moving forward. Here's our manufacturing plan. Here's our safety study plan, which is essentially important. Here's our clinical plan. Here's how we're going to dose the patient. Here's how we're going to follow up with the patient. And all these things the FDA looks over. In Cure Rare Diseases conversation with the FDA, our document was probably around 100 pages long. And it's very clear that the FDA reads every single page on that document and does a great job of providing really helpful feedback. It's a learning exercise for both us as well as the FDA as we introduce some of these new technologies. And it's very clear that the FDA is willing to collaborate and learn with us, which is really great for drug development. From there, Once we get that guidance and feedback from the FDA, then we trigger the really expensive part. And that's the production of the plasmid, or the therapeutic construct that we generated back here, tested in animal models here, discussed with the FDA. And now we're saying, okay, we're pretty confident that this construct, this version of we'll call it GNE, is the right one. And of course, you don't know until you get in human, but you use the supporting data of the animal and the cell experiments to make that decision. And then you trigger the GMP production. And again, GMP is the human grade version of the drug. So very, very expensive process. This is hundreds of thousands to millions of dollars. This is about 6 months worth of time. And so there's a long period here where now we've really interjected a significant uptick in the amount of time and capital that the project demands. And at the same time, we need to start to expand that mouse colony. So needing mice, rats, other experimental animal models, whatever it may be, pigs, for instance. It depends a lot on the disease, but expanding that colony then gives us the pool of animals to then test once the drug is finished in that pivotal study. So before and after the plasmid is produced and before the pivotal study begins, we have to put that package into a FedEx truck. And so that process is the virus production. That's AAV that we talked about before, the adeno-associated virus. What number you use is a little bit of art and a little bit of science. We did it as a data-informed decision. And really, the idea of doing lean preclinical development means that we want to take as many existing pieces of this approach as possible that have been used and have been in human before, and use those, because by using existing and known pieces, it helps to lower the amount of safety and efficacy testing that we have to do, which translates into quicker to the clinic. And so for cure disease, we made the decision to use AAV9. AAV9 has been used and continues to be used in trials hosted by Pfizer, Novartis with their spinal muscular atrophy drug for SMA type 1. And so using that known AAV type allowed us to move a little bit faster. And so ultimately this coalesces into a pivotal study. Now that study can take different shapes and different flavors, but more or less what it is the idea of safety first. Safety is paramount, both safety in animal. Do we see a crazy immune response in mice, although that's not exactly translatable to human? Do we see other concerning signs in mice that we think may translate to human? And also off-target analysis. Do we see, and this is important that it's done in a human cell line, not an animal, because humans and animals, although we do share common genes, they're not the same genes as a human has. And so that off-target analysis allows us to see in human cells, does this drug turn on or turn off genes that are really important? Genes we don't want to turn on, of course, are genes like cancer genes, and genes that we don't want to turn off, of course, are genes essential to the function of life. And so doing that analysis allows us to understand on a genetic level and on a in-life level, what do we see with the drug? And of course, no drug has I shouldn't say no, but most of drugs do have side effects, and those side effects go from a range. And at the end of the day, you have to look at the danger of the disease versus what are the side effects that we see, and make that risk-benefit analysis. And that's how the FDA looks at it. They say, okay, in the preclinical study, in this pivotal study, I'm just making this up, for instance, we saw heightened kidney problems. And so do we expect that they'd be able to be treated with an intervention after the gene therapy is administered? On the other extreme, do we see all these animals die? And that's a huge red flag, obviously. And so it's not that the drugs don't have risk. All drugs have risk. It's that we understand the risks and we understand the benefit, and we make that decision. Ultimately, the FDA makes that decision about pushing forward. And so that's a little bit of insight. And ultimately, we submit what's called an IND. This stands for Investigational New Drug, an IND application to the FDA. And that includes everything that's been done. That includes discussion around the construct, how it's made, how it's built, what sequences it contains, conversation about the animal model. Why did you pick it? What does it do? How did it work? Studies, PK and PD, pharmacokinetic and pharmacodynamic, just efficacy and biodistribution. Where does the drug go? What does the drug do? How does it engage with the body? How did you manufacture the drug? manufacturing of gene therapies is one of the most complex processes that exists today. And so with the manufacturing, the FDA puts a special focus on this to make sure that it's safe, and that we think it'll be safe. And then we bundle that all up along with the last study into this application that's multiple dozens, if not hundreds of pages long, and then submit that to the FDA. And the FDA reviews that, and they generally have 30 days to review that document, and then they make a decision, go or no go almost. So this framework, the idea that we can use this framework for tackling other monogenic diseases, I should say, is one that holds what we think at least a significant amount of promise. And so morphing into more of the focus on GNE, how this pertains to GNE is we're looking at using different constructs of the gene that our collaborators, some of whom I think are in the audience today, have generated. And the idea that we can figure out how we can choose the quote unquote best construct early on, because all that testing that we talked about in the last slide, all that testing depends on the fundamental of this construct. So it's quite an important question. Moving forward, again, back to that mouse study, we need to breed up the mice to have enough experimental models to dose. And you know, dosing one mouse isn't enough, because there's variability, there's all sorts of things that we have to have enough powered study, meaning enough mice to make a representative assessment to do that. And so in addition to starting to get the constructs squared away, selected and tested, we're breeding up mice to be able to meet the needs of that next study, the in vivo study we talked about a few minutes ago on that framework. And in that study, again, do we see therapeutic activity? Do we see the biodistribution of the drug? Do we see negative side effects anecdotally, although it's not a controlled study in the sense of it being a GLP study or a really well-controlled, well-regimented study? Do we see things that would concern us early on and that we want to go back and potentially change the construct before moving ahead? And can we replicate the study findings? Replication in science is so important, because we have to make sure that what we're seeing once, it holds for multiple studies. Seeing the same thing again really gives a lot of comfort to the effort. Once we've completed that initial study, then we'll go to the FDA again in that pre-IND meeting to get their feedback and guidance on the experiments we've completed, on the experiments that will be coming. And the guidance is helpful because it helps us to set and understand our expectations around the future, the defined studies that the FDA urges us to do to get into the clinic, expectations around manufacturing and what they want to see, they being the FDA, want to see in terms of reporting out and QC or QA, quality control. And also, as I mentioned, the perspective on the completed studies, you know, does the FDA feel as though this is the right animal model to use? And the way you engage with the FDA is all question-based. Does the agency agree, or does the agency think that the data supports the preclinical study? Something like that, more specific, but just to give you an example, that you don't go in and ask, hey, FDA, what do you think? You ask them very specific questions, they answer the questions, and then they'll also show their concern as well within the question as things move forward. So that FDA pre-IND meeting is a really important piece of the entire puzzle. Without FDA approval to dose, there is no dosing, and that's a really important piece to maintain public health, which is the FDA's job in the United States at least. Other countries have other regulators. The EU has the European Medicines Agencies. Individual European countries have their own sovereign regulator, but more or less in Europe, a lot of them funnel up to the EMA. Other continents have different regulators as well. So it's important to involve the regulators early and really treat them as collaborators. So once that pre-IND meeting is complete, we have a really good idea about the overall project. And what that triggers is the start of production of the human grade drug. So this is the GMP quality we were talking about earlier. So like I mentioned, it's the same drug used in that previous studies, assuming you didn't make any changes to the construct. It's just a higher quality. And GMP stands for good manufacturing processes, again, or practices. And that's to ensure that the drug coming out at the end is pure, is potent, is safe from the perspective of there's no contaminants, there's no things growing in there that shouldn't be. And I always think it's cool to see pictures of these, because we talk so much about theory and abstract that it's really neat to actually see a picture. So that vial right there that you can see is produced by one of our manufacturing partners. And that vial has the plasmid, the Amazon package, that does the therapeutic activity for my brother's drug. So that right there is one vial of my brother's drug. It doesn't have the virus yet. That's, like I mentioned, that's what's finishing up in June, but a cool picture to see. And then after that manufacturing is done, it goes from the plasmid to then being packaged into the AAV. That whole process, like I mentioned, takes about half a year when you're really pushing it. That will yield the material, which if FDA gives permission, will then go into human, but not before that safety study, at least how we've designed this framework. There can be different frameworks, but this is the one that we've built for this lean preclinical development. And again, this final safety study, it's pivotal study, as I called it earlier, is a much larger study. So generally the early studies will have 10, 20, 25 mice in them, whereas this larger study will generally have more like 50, 60, 100 mice. So you get more of a power behind it, a number of mice. And again, you ask similar questions. What type of side effects do we see? Do we see inflammation? Do we see crazy immune responses? Do we see death in the mice? Do we continue to see the efficacy, the target engagement, upregulating the gene that we want to upregulate? And how do we think those signals will translate into humans? Mice aren't humans. And at the end of the day, what happens in a mouse often doesn't, well, not often, I should say, but doesn't always happen in human. And so it's important to understand from the perspective of what that we see, do we expect to translate? And this study is done, I mentioned study quality before. Not only is there material quality, as we see with GMP material and research grade material, but there's also study guidelines in terms of the sort of regimentedness or strictness of the study. With pivotal studies, they often should be, if not always, under GLP criteria. That stands for good lab practices, excuse me. And so this is where everything's documented, right? So where did the material come from? What mice did it go into? What happened to that mouse? What tissue came from that mouse? Where was it sent to be analyzed? Who did the analysis? What did the analysis look like? All these very, very detailed questions that really help to inform and make sure that we're tracking every step of the way, so that if there's an issue or if we need to go back and make a change, that it's all been able to be tracked down to the individual mice, the individual operator doing the injection on the mouse, and so on and so forth. So very, very, very regimented, as it should be. So as we start to get to closing, kind of the more higher level perspective here, the FDA is rapidly adopting to the rise in next generation gene therapies and customized therapeutics, and so it's encouraging to see that the regulators, at least in the U.S., are really trying to keep up with the pace of innovation and the pace of science. It's a good problem to have that we're able to advance science as quickly as we have in collectively the last 5-10 years of gene therapy has become more of a common term. And so ultimately, though, the question of how do we pay for these things? NDF, a nonprofit, Cure Disease, a nonprofit, academic institutions, all these organizations, especially the first two, do a lot of fundraising through grassroots means, corporate partnerships. But really, the idea to make this sustainable is an important question. And ultimately, when we get sick with a therapy, with a disease or an illness that doesn't require a gene therapy intervention, you get strep throat, for instance, you go to the doctor, your insurance picks up the bill. And here too is what we're ultimately trying to advance the thinking of around payers or insurance companies, as you can equate the two. And we think that by showing success with gene therapies, whether they're a customized gene therapy, as with Terry, given that he's really the only one in the world, save for 1 or 2 other patients with that mutation, or GNE myopathy, the idea is that we need the system to step up and to support this wave of innovation. The regulators are stepping up as part of the ecosystem, and now too the payers need to step up to make this work sustainable. And ultimately, that's a conversation that's ongoing as we start to hear about things like value-based payments instead of cost-based. Value-based is the idea that this gene therapy will be applied, and what value does it bring the patient, right? Insurance companies don't want to pay for a drug that doesn't work, makes sense. And so what value do we see in terms of improved health to the patient and the patient community that would justify the price tag? And these gene therapies are expensive. Manufacturing is a whole different conversation and a much more complex conversation, but the cost and the effort to make these gene therapies is far, far more than a pill you might find in a bottle, for instance. And so with more complexity behind the therapeutics comes more cost, of course. And we as a society need to figure out, how do we deal with these things? How do we pay for very expensive drugs that are truly life-changing? And that's an open question that still doesn't have an answer yet, but definitely are driving toward an answer. So with that, just kind of want to sum up our conversation today before we start to shift over into Q&A. Just the conversation that CRISPR technology is a form of gene therapy, that for my brother, we use CRISPR to modify his disease. And the idea that we can take this approach that we've used for Terry with lean preclinical development and really generalize that to other monogenic diseases such as GNE myopathy is really exciting. The early work that we've already began to kick off at NDF as we pursue this lean preclinical development of gene therapy is really exciting. And so with that, let me turn my camera back on here. And
that was such a well-presented conversation, and I think really drilled down to the lay level where people who haven't been following along can really understand some of the jargon that we use. You explained it very, very well. Thank you so much. I'd like to thank also a couple of announcements I need to make is that today's talk was sponsored by the Beverly Hills Rotary. They are sponsoring our entire educational series this summer, so thank you to them. And next week, we'll be hearing from Dr. Kelly Crow, drilling down further into what Rich talked about today in search of a biomarker. So looking forward to that chat. I'm going to introduce Dr. Angela Leck to moderate the Q&A. But Angela, before I let you address some of the questions, and as patients and listeners are typing in some of their questions into the Q&A, I wanted you to perhaps address for some people that have had this question, how this particular effort and our support of this approach and our interest in this approach can augment, dovetail, or be in harmony with some of the other more longstanding approaches and efforts towards gene therapy, sort of the next gen. So if you could address that for me, that would be my question for today.
Okay. Well, the NDF is, very much wants to learn from the lessons that Rich has gathered from his journey in order to help. The idea is, simplify or to expedite, the process of getting a gene therapy into the clinic for GNE. But obviously, the lessons learned can't be entirely applied because Rich's journey is in designing a gene therapy for his brother, for a small patient population. And GNE, even though it is a rare disease, it's relatively a larger in comparison patient population. So there is going to be some differences there, which in a second, I'll ask Rich what he thinks some of those differences may be in having a conversation with some of the regulatory bodies. So I've been working with Rich now since, as you mentioned, 2018. And I think, and in Rich presenting this sort of case study to the NDF board, the NDF is now interested in pursuing a similar path. And NDF is looking to work with as many scientists in the research community as possible to help expedite the preclinical data generation towards an IND. And I'm honored to be helping manage those collaborations and reaching out to different scientists about how they can help with this effort. So I've already reached out to Dr. Stella Rosenbaum and Marianne Huizing that are already involved in the planning stages of this effort, such as with construct use, which mouse model, and we recognize, that different scientists have different expertise that they can bring to the table in the field. and I, want to strongly suggest that we all work together to bring this forward as kind of one effort. And maybe I'm being too optimistic, but I'm excited.
One of the things, I think you're right, Angela. One of the things that at NDF, it's a philosophy, it's a belief that we have an obligation, not just pressure, but obligation to approach any solution suggested to us that's feasible and let the chips fall. But our job, my job, is to line up door number 1 through 5, 10, 15, if possible, and wherever the answer comes. But of course, always with the understanding that we are collaborating, that people don't work in silos. I don't think that's good for the industry. I don't think that's certainly not good for the patients, that we build on top of the information that we all gather. And I just wanted to hear your explanation, and I'm curious to hear what Rich has to say about that.
It's such a, with rare diseases, it's really important to try to get as many people working together as possible. And you're right, I don't, people working in silos, that's just going to slow everything down. There needs to be a concerted effort and money thrown towards a concerted effort in order to accelerate this thing. It's whether we take this we are the ones that take this to the clinic in collaboration with a clinician, or whether we take this to a stage where we generate enough preclinical data that makes GNE an attractive disease and lowers the risk for a pharmaceutical company to then step in and then take the reins from us and then develop this program further. That remains to be established, but we don't know what the end outcome will be, but I feel that this is the right path.
Right. And again, thank you for underscoring that, because we are looking for that level of collaboration and out of the box thinking, as well as traditional understanding of how things work. So I will turn it over to you. I know you have your own questions. Thank you so much to both of you. I'll be listening with great interest. Thank you.
Okay, great. You know what, last week I had problems seeing the questions, and this week I don't see any questions at all. Can you see any questions, Rich, in the Q&A or in the...
No, but I think one of Lale's first questions that might be helpful to tackle is the question about how does this differ from N of 1 to N of several more. And I think it's important for people to know that the difference there is that the FDA looks at a drug going into a patient or a patient population all through the risk-benefit analysis. So if a drug is going into one individual, there's a different risk profile than if it's going into 10, 100, or 50, 1,000 people. And so how they change that assessment could be longer testing. So our safety study for Terry is a 3-month safety study. So potentially the FDA for a bigger population, requires a 6-month safety study, or a safety study with a larger animal species, dogs or NHPs, non-human primates, that could be possible. So the idea is that it's a framework, that it is flexible enough to really stand under the muster of larger rare diseases, still rare, of course, but larger than 1 or 50 or 1,000. And so that's one of the ways in which there could be some variability, of course.
Yeah. Thank you. And yeah, I just wanted to thank you again for sharing your incredible journey with us. And personally, I want to say that the work that we've done with CRD cure rare disease has really been one of the highlights of my scientific journey. And, the thought that Terry, your brother, may be dosed in 2021 is totally mind-blowing to me. Given that CRISPR has never been used systemically to address a muscle wasting disease in human, we have to remain cautiously optimistic, but ultimately we have to respect that it is the patient's choice. It's once the FDA approves it, it's Terry's choice. He's an informed adult, and it will be his legacy if he chooses to take this therapy, which I greatly respect and admire his bravery. So, but going back to GNE, we the NDF pretty much very much wants to learn from the lessons along the way, and seeing how a nonprofit can be the sponsor of an IND and work with all the important stakeholders in the field, such as the patient community and all the different scientists, and really bring everyone together, because it is such a rare disease that we can't each work on our own different thing. We have to work together, and it needs to be centrally coordinated by an unbiased sort of organization that has its priorities in the right place. So
yeah, agreed. And I see we've got a question here. Appreciate the comment, Tara. And as to the other question, appreciate that as well. Is there some takeaway that we can learn to move forward at a similar pace as we've gone? The answer is collaboration. Very, very clear answer there is breaking down the silos between the different functions. So Angela and Monkol represent our early stage, our what we call discovery part, and they are who generates the construct. Having that construct translate to being dosed in animals, being manufactured, are things where you need a team all on the same page, so that an expert in manufacturing, actually this happened to us, we were using a certain, without at risk of getting a little too complex, I'll try to keep it simple. We were using a certain antibiotic involved in the drug, simply not necessarily as a function of the drug, but as a supporting mechanism. And the manufacturing crew went, actually, that's not going to work for our manufacturing process. So we changed that at an early stage to avoid downstream delays and disruption. And so, you can see that it's just getting the right people in the right room talking, and really pushing aside the silos and the siloing is by and far the large way you can do that. And folks that are listening, you have an active role in pushing that collaboration. Having the constant question of how are you collaborating, and holding the different stakeholders accountable to ensure that collaboration will happen is really important.
Yeah, and Rich mentioned we were involved in the early data generation, but we don't have expertise, and no lab has the entire toolkit required to get from the beginning to the very end, which is the clinic. So we need to recognize what our strengths are, and then pass the baton on to another team that has that expertise in safety study and toxicity study, and in whatever. So I have been talking to a lot of the different researchers in the field to identify what their strengths are, and then to tap into that strength to see how they can accelerate this process and help with the combined effort. So yeah, if you're a scientist and you're on the call and I haven't contacted you, just you'll be hearing from me. So we have a question on the Q&A. If multiple siblings have the same mutation, could they all receive the same gene therapy? I can quickly jump in and say, with the gene therapy that we are planning, which is a gene replacement therapy to give an entirely healthy copy of the gene, the answer is yes. It should be applicable to all the patients, regardless of the mutation. So there's more questions for you, Rich. So Imad, Rich's story is inspirational. It's amazing to see how you started. Oh, that's not a question. What can we learn to move forward at a similar pace? Well, you've covered some of that. Is there anything else you'd like to add?
No, I think Imad had a follow-up question as to the answer. What can we do to ensure that the collaboration happens? And I think this is, where patients play a great role here is, going to the conferences and asking the challenging questions. More or less kind of thinking on the fly here. So I'm sure I'm not going to get them all, but what are you working on? How does that translate to helping patients? Who are you collaborating with? Who could you collaborate with? I think asking the questions is a really important piece of advancing the overall field, and it's up to patients to hold researchers and clinicians accountable, because ultimately it's patients like us at the end of the day who have the most to lose. And so, we have to push past this idea that publications are really the ultimate goal. We like to joke that it's a great time to be a mouse because mice are being cured, of these diseases and then the findings being published, which is great, but that's only the first step. We have to take that step further to say, okay, that's great. There's a publication, the mice have been cured. Now, how do we translate this into the next step of development? How do we get this into the clinic? And those are some of the questions that encourage folks to really think about and ask to make sure that things are moving forward. And then there's another question too, Ange. I see on your proposed framework, it would take 3 or 4 years to get the IND into treatment. Will this be similar for GNE myopathy? And what about the over 260 GNE mutations? So do you want to take that one in terms of the—
Sure, yes. So I think that is a reasonable time frame. It's really hard to foresee all the steps, but we discussed this amongst the board members. And we do think that that is a reasonable time frame, less than 5 years. And as to the specifics, it really depends. I mean, the FDA, they changed their mandate, whether they require, non-human primates or large animal study. They're constantly changing it depending on what's happening in the field and the latest, gene therapy human trial results. So it's really hard ahead of time to foresee these mandates. So, but I think that less than 5 years is a reasonable bet. But yes, as I mentioned in the previous question, it would be able to, it's not a CRISPR-based approach. If we just restoring the gene, healthy copy of the gene, then that would be applicable to all patients. Yeah.
Yeah, I think just building on the question as well, the 3 to 4 years, it's tough to predict, right? We, in some ways, we're very fortunate because the experiments worked. The question to ask is, well, what if the experiments didn't work? And at what point didn't they work? And that really informs a lot of the timeline. I think, if we can help to mitigate the chance that experiments or efforts such as manufacturing don't work by getting the group together and on the same page and the silo is broken down. But there is a function of, there's a function of luck in this case that, an experiment goes well, and that's great. And we're able to replicate the results as we were able to do, and that's great. But there's also the case that, okay, maybe the construct doesn't work the first go around and we have to modify it and then redo some of the early stage work. And, that's months, that's not years. But that's why we try to de-risk a lot up front, so that once we get committed to that manufacturing of the human grade drug, that 6-month time period and a lot of money, that we don't have to do that twice, because that's where things go from months to years, and having to replicate. And to Angela's point about, the FDA is very privileged from the sense that they get to see everything. We see what we do and what our colleagues choose to share, but the FDA is able to see experiments from companies, from academics that have chosen to meet with them, and they take that perspective and they apply it to programs so that we're able to make the best decisions, even though we don't know the specifics of, for instance, I'm just picking on Pfizer today, no particular reason, but everybody knows them. The FDA is able to see what has Pfizer done, and they're not going to say, oh, Pfizer did this, do it this way. They're going to say, well, we want the analysis done so that these are the endpoints, because then they'll be able to see, okay, if there's a problem, we should be able to see it in this regard. So hope that answers the question.
Mm-hmm. What can we ensure it happens? Well, short answer, funding.
Yeah, funding involvement, I'd say. Yeah, I think we
grassroots fundraising.
Yeah, I think this is just a patient family perspective, but I think for most of drug development, it's sort of been, the researcher clinician here and the patient there. It really needs to be an equal partnership, because each group brings an incredibly important aspect to the table. The patient, of course, brings the disease, and oftentimes, samples are very hard to get, skin samples that can be transformed into muscle and other things. So the patient brings the perspective of the journey, the actual samples that enable research. The researcher, of course, brings the know-how, the construct. The clinician does the dosing and the patient care. So each one's very important, but I think we are out of time now. Yeah. Well, hopefully, I think we got to all the questions.
I think covered everything.
Thank you so much, both Rich and Angela, for moderating this session. Rich, your information was incredibly helpful. I'm getting a lot of texts and notes and emails from patients thanking you. And this, of course, will be posted on our website at curegnem.org. by Monday, and anyone will be able to download it and watch it again, as is the case with all of our videos in the Learning Library. Thank you to everyone again, and we'll look forward to seeing you guys next week with Dr. Kelly Crow discussing biomarkers. Thank you to everyone. Have a good rest of your day.